Right, we're now going to move on to one of our keynote presentations, actually a pivotal presentation for the whole symposium. And I want you to quieten down. And I want you people at the door. All this socializing. You're here to study. So, but for those of you who haven't heard Peter Visscher present before, you're in for a real treat. Peter is one of the most influential figures in modern statistical genomics, statistical genetics. And we all know about Darwin, and we know about the modern synthesis last century, but I think Peter's one of the leading founders of what's happening now in the way we're kind of really understanding the genetic architecture of human traits. And that underpins human disorders that Naomi presented this morning. Peter is a named co-investigator on the Niels Bohr Professorship. He's a co-director with Naomi and Jian Yang of the program on complex traits at the University of Queensland. And Peter's lab specialises in developing methodology. He's really contributing to the toolkit and telling us what we can expect as we unravel the genetic architecture of diseases and traits. He's one of the world's most respected and prolific researchers in the statistical genetics. He's much awarded, and I'm going to embarrass him a little bit now. So he's a member of the Australian Academy of Science, and probably many others I don't know about, but there's two that have occurred in the last few weeks that give you some index or metric of the influence of this guy on the field. Peter was recently elected a member of the Royal Netherlands Academy of Arts and Sciences. And a few weeks ago, he joined Darwin and Newton when he was elected a Fellow of the Royal Society. That's a fantastic recognition, and I want you all to join with me and congratulate Peter on that. Respect. So I'm going to ask Peter to give me all my references from now on. But Peter, like Naomi, is really we're wanting to build more links between Denmark and Australia, more links between the iPsych and Peter, Naomi, and Jian's group. We want to have more of that cross-fertilisation, where our staff go to Brisbane and the Brisbane Peter and Naomi staff come here, and we get that creative energy that is happening in Peter and Naomi's group. You heard that from Naomi's presentation, where various people from different backgrounds are able to sort of crystallise new ideas. So Peter, I welcome you to the podium, and we look forward to your presentation about big data and complex traits. Thanks very much.
Thank you, John. Can you hear me okay? I'd like to echo Naomi's words that we're very proud of you, John. And I think, as a scientist, I'm more proud of you than I am of Mary. But before you go on, it's wonderful to be at this symposium and wonderful to be back in the Kingdom of Denmark, famous, of course, for Hans Christian Andersen, Niels Bohr, and, last but not least, Lars Ulrich. For those who like Naomi, who doesn't know who Lars is, he's the drummer of Metallica. So I'm going to talk to you also about unpublished work. I think this paper is likely to come out next month in Nature Genetics. And I'll start by acknowledging at least the authors who are listed at the front and back end of the paper. But just to, in case you get a wrong impression that the other 100 authors in the middle don't matter, I'll list them all here, and in fact there are Danish authors in here as well. I think there's a few authors from Copenhagen, but don't ask me exactly where they are. So I've got a whole bunch of slides trying to justify to you why it's interesting to work on a trait like educational attainment. And then both Naomi and Jerome actually mentioned educational attainment on a number of occasions with respect to psychiatric disorders, but I'll still go through my justification. So educational attainment, like human height, I think is also a kind of model trait. Its heritability is similar to that of major depression, for example, and we know there's both genetic factors and environmental effects that contribute to variation in the population. Educational attainment is extremely polygenic, as we know from previous publications, and is very easy to measure. So we can get large sample sizes, and the larger the sample sizes, the more we can learn, and the more the data can generate hypotheses that we can test subsequently. Educational attainment is also associated with a number of important outcomes later in life. Not just how much you're likely to earn and other socioeconomic outcomes, but also your likelihood of getting a number of diseases and disorders later in life and how long you live. So from that point of view, from a health point of view, it's an important trait. Here's a link to psychiatric disorders. Actually, two papers I picked up yesterday with help from Naomi, because she had them referenced in a paper with Wouter Perot. On the left is a meta-analysis from 2003 showing, and here's the main results, that every additional year of education reduces your probability of major depression by about 3%. At least that's the conclusion from this paper. This paper is a different paper. It summarizes a longitudinal study on adolescents, and that suggests that if you have depression during adolescence, that it's actually a risk factor for, I'm having to look here to look what's on the slide, it's a risk factor here for dropping out early and for not going to university or tertiary education. So two different sort of directions there. Apart from these observational studies, we also have genetic correlations, and a number of them have been mentioned previously by both Jerome and Naomi. This is a paper from Bullock-Sullivan 2015, who introduced this method called LD score regression, where you can take summary statistics from genome-wide association studies, from different studies, and estimate a genetic correlation under some assumptions. So here is years of education, and a sort of reddish color means a negative correlation, and it's negatively correlated with a bunch of things that are not good for you, including smoking and risk of some diseases. Also, in a previous publication on educational attainment, there were genetic correlations associated with a number of neuropsychiatric and behavioral traits, and here you can see that genetically having an increased predisposition to education, whether that's cognition or other traits that are associated with it, reduces, it seems to be protective for Alzheimer's and for neuroticism. And according to this publication, is positively correlated with bipolar disorder. A number of the previous speakers have mentioned Mendelian randomization, and this paper was also mentioned, that came out this year. I have to emphasize that both John McGrath and I are co-authors on this paper, and here we try to estimate what I call putative causal associations between exposures and outcome. And here I've picked up the two tables that, based on very large sample sizes, UK Biobank and the JERA cohort, looked at the putative causal association between years of schooling and outcomes in terms of disease. And as you can see, years of schooling appears to be protective for a whole range of diseases here, cardiovascular, type 2 diabetes, etc. And even when we count up to a count of all the number of disorders that people in the UK Biobank actually mentioned, we're getting a protective effect just if you sum them all up. And this is, doing this for case control studies, we find also protective effects here for again, coronary artery disease here and rheumatoid arthritis, not clear why that would be. And we found here that apparently, according to these data, years of schooling is actually a risk factor, or the the genetics of that. Well, this is supposedly be a causal relationship that is genetically correlated with years of schooling being a risk factor for autism. So I hope I've convinced you that studying educational attainment is not a bad thing to do. So I'm just now going through the previous two studies that were published and what we found, and showing to you, and I hope to convince you, the power of sample size. So in 2012, David Cesarini, Dan Benjamin, and Philip Kullinger got together and established the Social Sciences Genomics Association Study, or whatever it's called exactly. And I was fortunate to get involved in their endeavors. And they realized that in particular in behavioral and cognitive literature, when it comes to genetics, that there had been a lot of problems in the past with experimental designs, with sample sizes, etc., and they wanted to do it properly. So for the first educational attainment study, we got together a sample size of 100,000, which at that time, this is 2012, paper published in 2013, was very large. We harmonized according to some international scales, the education years, and also had a trait which was whether or not you went to college. And then performed this kind of meta-analysis, and we found one genome-wide significant association for edu years and 2 for college. And actually, genetically, these traits are very highly correlated. So that's 3 hits, which is not very much for 100,000 discovery sample size. And in the replication, we had 25,000 on all 3 hits replicated. And this is the Manhattan plot, as it's called. So here are all the SNPs, and here's the strength of evidence for association. And this line here, and I'd like you to remember where it is, because we'll see some other plots where you might not be able to see it anymore. This is the line of so-called genome-wide significance, and here are the three hits. And together, and this paper was published in Science, because we argued this was probably the first robust associations between a cognitive phenotype and genetics probably ever reported. Some people may disagree, but that's what we argued. And together, these variants, if you say how much variation in the population do these 3 variants explain, hardly anything, much less than 1%. However, we realized, okay, this is the polygenicity, and this is what educational attainment looks like. We just have to ramp up the sample size. And fortunately, educational attainment is an easy byproduct of lots of very large epidemiological studies. So again, it's easy to get very large sample sizes. So come 2015, paper published in 2016, we had 63 data sets. It's now the second study with a sample size of nearly 300,000. Again, we followed with a pre-registered analysis plan, which I think is very good practice. And it was essentially the same as for the first study, but we decided, because these traits were so highly correlated and educational years are kind of easier to measure and harmonize across countries, that we just focused on the years of schooling. We found 74 hits from that study. And when the paper was being reviewed, the first release of the UK Biobank became available. So it seemed crazy not to use that. And we thought at the time, do we need to add that and have a whole new discovery phase? But we decided to keep it clean and only use it for replication. And interestingly and unusual for many of our studies, the replication record was almost better than we expected just by chance. So 72 out of the 74 lead SNPs had a consistent sign. 52 of the 74 in the UK Biobank were significant at the 5% level, and also with the same sign. And 7 actually reached genome-wide significance on their own, just in the UK Biobank. Again, this is actually also shows not just what a powerful resource the UK Biobank is, but how much sort of value for money it has in terms of the amount of information per data point, which is much larger for many traits than it is for meta-analysis. And here's the Manhattan plot for that study, and as you can see, the line here is now many towers here, 74 lead SNPs. And this was published in Nature in 2016. And just to show you how good this replication was, this is our study where what's called meta-analysis here is excluding the UK Biobank. So this is the meta-analysis from which we found 74 SNPs. And here are the estimated effect sizes, the regression coefficients for years of schooling from the UK Biobank. They are in blue. And you see it's remarkably consistent there. Okay, so that was sort of background, and now we go from 2016. You see how quickly this goes, and fortunately, we also see that for genetic studies in psychiatry, that these sample sizes are becoming extremely large. So This time we have 70 data sets with a sample size of 1.1 million, which I think is probably the largest GWAS to date, although there may be another one which is slightly smaller, which is already published. So basically we just took the published results of the second study and added a bunch of new data, in particular the UK Biobank, the full UK Biobank set, and data from 23andMe. We followed essentially the same analysis plan as in the previous study, and we found now nearly 1,300 hits that were genome-wide significant. Just to give you an idea how small these effect sizes actually are, the median effect size of these significant hits are about 1.7 weeks of schooling per allele. And now you might think that is not small, but the standard typical phenotypic standard deviation for educational attainment in the population is of the order of 5 years or so, which is very many weeks. So very small effect sizes, and therefore you need these huge sample sizes to pick them up. And this is now the Manhattan plot. I mean, look at the scale. Here's this line that I was mentioning before, where only 5 years ago we only had 3 hits that were just about above this line, and this is what you get when you have 1.1 million people. And this shows you the replication of the SNPs that we presented in the second study, and this is a replication, obviously leaving those data out. So this is an independent replication. Here are the effect sizes, which obviously have been ordered to the increasing effect sizes here in the second study, Educational Attainment 2, published in 2016, adjusted for Wiener's curves in some, which you can do making some assumptions here. And here are the replication effect sizes. As you can see, nearly, apart from these few here, nearly all of them are consistent in sign. And on average, the effect size is, well, as you can see, it's clustered around what you'd expect the result to be in terms of, on average, the same effect size. But of course, there's variation around that, and some are not very significant, but most of them results are very significant. So, kind of robust replication. I think for the first time ever we did a powerful enough analysis on the X-chromosome, which curiously has been neglected in a lot of genome-wide association study, even though many of the SNP chips have had X-chromosome data for a long time. So we got X-chromosome data from our own study and from UK Biobank and from 23andMe, I think, because lots of the smaller studies had just not produced that at least not in time. And this is just a simple, chromosome-wide plot on the X chromosome. We found 10 SNPs on the X in total. We calculated the genetic correlation for males and females on the X, and it's essentially 1. So there's nothing special in terms of the X chromosome in terms of effects on males and females. So no sex by genotype interaction or no evidence thereof. And although I'm not showing you this, we also developed a way to look at the effect of dosage compensation, which is the fact that females have 2 X chromosomes and males only 1, and females compensate from that by essentially expressing only 1 of them. And dosage compensation has a consequence on the amount of genetic variation that you might expect in females and in males. But overall, the amount of variation contributed by the X chromosome is very small. So these are just putting all the results together, including the X now. So on the left-hand side here, we have on the X-axis the length, physical length of the chromosome. See here, and here the heritability that we estimated on that chromosome, and here the number of lead SNPs that we get from the GWAS. And first of all, the thing to notice is, which we've seen from many traits, including psychiatric traits, there is a strong correlation between that longer chromosomes explain more variation, and therefore you find more genome-wide significant associations on those hits. It does seem from this that the X chromosome, which is coded here, is a bit of an outlier, suggesting that relative to its physical length, it seemed to explain a bit less variation. And there are fewer hits. And it is not entirely clear why that is. First of all, the X chromosome, what we left out from the X chromosome is some regions which are the pseudo-autosomal regions, and the SNP chip that we use for this is mostly UK Biobank, which I think had not all areas of the X chromosome really well covered. Interestingly, if you do the x-axis slightly differently, and it could also be that just because of this dosage compensation that you get less variation on the X chromosome, because the amount of variation you expect on the full dosage compensation in females is actually less than that of in males. So that's also a possible explanation. However, when we calculated from the SNPs that we had on all of the chromosomes, some kind of effective number of independent SNPs, which just means taking account of the correlation structure between all the SNPs on the chromosomes, or linkages equilibrium, the results fall much more in line. Including, so the X is not an outlier after you account for the stronger LD that there is on the X chromosome. This is just an observation, and there are many possible reasons for this, but we don't know and just present the results. One of the things you can do with these large sample sizes, when you also have access to family data, is you can compare the estimates that you get from the population with the estimates you get in families. And I hope to convince you that that's quite an interesting thing to do, because you might find something you don't expect. So we got about 22,000 sibling pairs, so just brothers and brother pairs, sister pairs, brother-sister pairs, for which we had SNP data and also educational attainment data. These are from a number of cohorts, including the UK Biobank itself. So we looked for just concordance of the sign of the effect size. Obviously, we excluded these data from the discovery, from the 1.1 million. We looked at the concordance of the sign and also calculated what we expected that distribution of the sign concordance to be, because there's always standard errors on the estimates, like sampling variation. We also estimated the effect sizes of the genome-wide significant loci, what they are in the families, and we estimated some kind of single parameter which you can think of as how much, what's the ratio of the effect sizes, or strictly speaking, the variance explained of these SNPs within families versus the population. So here are the results of the sign concordance. So what I have on the left here is what you expect under the null hypothesis that all the population-based genome-wide significant loci are false positives. Then clearly you're getting an average of 50% sign concordance, but you get this distribution around it which depends on the sample size of your within family study. So, and as you can see, just by chance you could get, you know, 0.5 something or other just by chance. Then we go all the way to the right-hand side, and the green one is what you expect the sign concordance distribution to be under the hypothesis that these really are SNPs that are associated with educational attainment, and we adjust for the Witness curse. And the Witness curse in this case is just ascertainment or selection bias, that out of the 1.1 million, out of the 10 million SNPs or so we looked at, we pick up 1,300. These are genome-wide significant, but because you do this selection, their estimates are still slightly biased upwards. So when you adjust for that, we expected a sign concordance of, let's say, 0.75. We can do a third one, is that under assortative mating, we actually know, just from theory, which goes back 100 years to R.A. Visscher, we know that the effect sizes within families are actually smaller than they are in the population. So we can adjust for that as well, and that's the blue distribution here. So under the null, that all effects are spurious. Under the alternative, that there's no assortative mating, and under the alternative, there is assortative mating. These are the results we obtained, which, as you can see, are in no man's land. They clearly deviate, they're much more than you expect by chance, but they're not what we expected. And here are the estimates from this parameter I mentioned, which you can think of is what is the variance explained of the SNPs within families as a ratio of that to the population. And here are all the different studies, but let's focus on this one. When we don't do a correction for assortative mating, it's about 50%, and when we adjust for assortative mating, it goes up a bit, but it's still way shorter than 100%. Just to convince ourselves that we weren't doing anything wrong and that we had a comparison, we also did this for height. And when we do this plot for height, the estimates are very close to 1, saying that if you estimate an effect size within a family, within families, so that the analysis is simply essentially regressing the difference in the phenotypes of SIBs on the difference in the genotypes that they have. For height, the estimates are much closer to 1, suggesting that the within family estimates are very similar to the population ones, but not for educational attainment. So the question is, what's going on? Let's think of a few possible explanations why these effect sizes can be so different. The first of all is Witness curse, that the effect sizes are hugely overestimated, the effects we estimated from the 1.1 million people. This is likely that we have Witness curse, but we try to account for that quite rigorously in the analysis. How about population stratification, which is a confounder which could also lead to spurious results or overestimation of effect size? Possibly is this an explanation, but we had quite stringent control on population stratification, and we don't think that's the likely explanation. What about assortative mating? Yes, there's likely to be assortative mating on either educational attainment itself or something correlated with it. But as I showed you, we try to account for that in analysis. So the most likely explanation we think is essentially not a G/E interaction, but a correlation between the genetics and the environment. So that parents who have on average a higher predisposition for educational attainment, let's for now call them smarter parents, invest more and provide more environment, nurturing environment to the children for them to have a higher educational attainment. And the beauty of these within family analysis, because you're looking at the difference between the siblings, those effects go away of the parental nurturing. Indeed, there's a paper from the Decode group, the Iceland group, that came out in Science, now I can't remember if it was this year or last year, which actually modeled this directly, because they have all this beautiful pedigree data where they can follow the transmitted allele from parents to offspring, and they know which is the non-transmitted allele. And they modeled this in a clever way, and just to show you results for educational attainment, they used the polygenic predictor, of which we've heard about a lot this morning, and if they just looked at the transmitted allele, so this is a bit like the population-based analysis that I showed you, it explained 5% of the variance in educational attainment. If they adjusted for the fact that the non-transmitted allele also had an effect, and that's the kind of environmental or nurturing effect, that went down to 2.45. And remarkably, this ratio is close to the 0.5 that we also observe with our sibling data. So I don't call this a bias, because in genetics, if you go to a genetics textbook, G/E in most analysis, the genetic analysis you go by, I'm not talking about G/E interaction, usually goes to the environment. GE correlations, classically, are seen as genetic effects. So I wouldn't call them biased, I'm just saying, look, this is what you get when you estimate the effect size within family versus in the population. If in the population as large, there's always this correlation between your genotype and the environment, that's what you get, and that's what we observe. Heterogeneity was also mentioned, and indeed, just like for major depression, I think educational attainment, partly because we we combined data from many different sources, we found evidence of heterogeneity. We did tests on the 1,300 lead SNPs, and we can reject the null hypothesis of a homogeneous effect across all these different studies. And we also try to estimate genetic correlations between pairs of cohorts. Unfortunately, many of the 60 cohorts that we used had actually quite small sample sizes, which means that the standard error of the estimate of genetic correlation is very, very high. So when we did all these pairwise comparisons across all the pairs, we get an average of about 0.7, but still with a large standard error. But clearly the evidence is that it's significantly lower than 1, which I think shouldn't really surprise us. We're comparing here some data from completely different educational systems. From some countries where people pay for education, for others like here, I presume, where education is free, etc., etc. So now I'm going to go, so that was essentially all the GWAS, the estimate of effect sizes. Now I'm going to go on what you can do with these data, and again, talking initially about genetic predictions. So we created from these 1.1 million people, we created a prediction. You can do, there's many methods that are out there to do this. For this particular paper, we use this method from Alkes Price's group in Boston called LD-PRED, but there are other methods that perform very similarly. It's kind of a Bayesian approach. The details don't really matter because the proof's in the pudding here, in that you can see how well these predictors do when you go to out-of-sample prediction. The prediction samples, which were not part of the discovery, were 2 samples where there were lots of phenotypes available, one called AdHealth and the other the HRS. And for this analysis, we only used about 1 million or so SNPs, which are quite common, that would capture most of the common variation in the human genome. And we calculated these scores, which I think here are called polygenic genetic scores, because to talk about risk for a quantitative trait, it doesn't really make a lot of sense. So how do we measure how well our predictor works? Well, very simple. We have a phenotype in the new samples, and we have a bunch of control variables or covariates, and we fit a model, a regressive phenotype on the control variables without the polygenic genetic score, and then we rerun it by adding the score and look for the difference in the R-squared, for example, the regression R-squared, or for binary phenotypes, some kind of pseudo R-squared from a probit regression. And we also calculated confidence intervals using bootstrapping. So here the main results, I've summarized all of them from the previous studies as well. So on the x-axis here I have the sample size, which goes back from the original study of about 100,000 all the way here to 1.1 million. And this is the incremental variance explained just by the genetic predictor. So initially the paper where we only had 3 genome-wide significant loci, when you take all of the SNPs, you actually still explain about just under 3% of the variance for this. It's color coding for the two cohorts, and 4% for the AdHealth study. Here the results from the 2016 paper, now based on either on 300,000 individuals, which are these 2 points here, and when we added a UK biobank, remember that came in during revision, but we could add that in as part of the predictor, and that explained about 7. As you can see here, 6, 7, 8% of the variance. And now, with 1.1 million, we're up to 11, 12, 13% of the variance explained. This is quite remarkable if you think about that these 3 variants that were identified only 5 years ago together explained 0.1% of the variance or something, right? So what can you do with this? Well, you can see how well it does it predict stuff in a new sample. So here is an example of the trait whether or not you completed college. So this is again, this is heavily phenotyped data sets. Here we've just split the predictor into 5 groups with equal sample size and look at the mean prevalence of college completion for the individuals who are who have scores in, say, the lowest 20% or the top 20%. And you can see for both these studies a nice linear increase, and also that it actually predicts a not unsubstantial difference between the highest 20%, which are about 50% completion rate, versus the lowest, which are about 10%. You know, these are big differences when you accumulate this kind of information. You can also look at educational achievement, how well kids do at high school, for example, or other cognitive phenotypes they may have measured. So here again we have the education years itself, for which I showed you the results, which were 12 and 11% or so. Here we have or not whether kids completed high school. The predictor explains about 5% of the variance there. Whether they completed college, I showed you that already. Here, interestingly, for the AdHealth study, which is an adolescent health study, you find the school GPAs that the genetic predictor here explains about 6 to 8% in the school GPAs of these kids. And all these results are obviously highly statistically significant, because these are sample sizes of many thousands, and you don't need to explain a lot of the variance to get a significant result there. How does the genetic predictor compare to non-genetic predictors, or I should say to predictors that may partly be also contain a genetic component, but are more easily measured, for which you don't need genetic data? Well, if you take the parent average for education, it does a lot better than having a genetic predictor, which probably shouldn't surprise us, because parental education captures both the genetics of the parents but also their environment. So that has an incremental R-squared of about 20% or so, as you can see. When you take mom's or dad's education on their own, they perform similarly, slightly better than a genetic predictor here. But if you look at predictors like household income, which is used in the social sciences, then it doesn't do as well as your genetic predictor or marital status, for example. We also recently developed, and I was sort of part of this development team, some predictors which use more information than just the associations of SNPs and the trait itself. It uses information from traits that are correlated with it. Because in a way, the more data you use, if traits are correlated, you should be able to borrow information from other traits to make better predictors of the SNP effects and get better prediction accuracy in a new sample. So this is when, this is this method called MTAG, published in Nature Genetics, and there's another paper from Robert Meyer also that came out in 2018 in Nature Communications, which also has a multi-trait predictor, does it slightly differently. But all these methods can be used just by exploiting all these summary data from GWAS that are available in the public domain. For example, on LD Hub that Naomi mentioned this morning. So for education years, we okay, we had data on years of education, but also from 23andMe, data on cognitive performance, which is in the public domain. And from 23andMe, we also had data on about 600,000 people or something from self-reported math in the highest math class that they attended. If we look at a multivariate predictor for education years by adding these other excuse me, adding these other traits, we're getting a modest gain. And that's shown over here at about 0.3 to 0.6%. However, it gets much better if you want to predict, for example, cognitive performance, and then you add educational attainment as a predictor, because that has a much larger sample size. So then you get increases of 2 to 3%. So depending on the trait that you work with, for example, and this has been shown also in publication, if you work on schizophrenia, you want a best predictor for schizophrenia, and you have data on bipolar, it gives you a little bit, but the data sets of schizophrenia at the moment are much larger than for bipolar, so the gain is quite modest. However, if you want to increase your prediction accuracy for bipolar disorder, you could borrow information from the GWAS on schizophrenia. And you should get an increase in prediction accuracy. So here's just some examples of what you might want to do with these predictors, and this is partly geared towards the social sciences, but I would hope that that might be relevant in psychiatric epidemiology. So if you take one of these predicted values from an individual, so an additive genetic factor, if this additive genetic factor explains 10% of variance in the population, then it's very easy with a very small sample size to find something in your own experiment that's correlated with it, if that's what you want to test. In fact, this calculation suggests you only need 75 individuals. I'm not an advocate for small sample sizes, okay? Don't get me wrong, but this is just how the sums come out. And in principle, if you have small studies, for example, in cognitive psychology or whatever, usually small samples, because they're usually university students, like 30 treatment, 30 not. It would be worthwhile basically to genotype all these individuals, because you get, first of all, you control better for potential confounders, but also you gain power. Or you could reduce sample size, but again, I'm not an advocate for small sample sizes, don't get me wrong there. And here's another, two results on methodology that John asked me actually to talk about, but the methodology is in the background here, because I think the results are just more interesting. But here are two results from two published papers, one's published, one's under review actually, but it's on bioRxiv, where you can also look at educational attainment, with a different purpose in mind. So here's a study published this year in Nature Genetics from Jiang Yang's group, and we were involved in that, which is trying to find evidence for natural selection, past natural selection, on loci that are associated with particular traits. And one way to look at that is to look at the relationship between allele frequency of markers, of SNPs, and the cumulative amount of variance explained. If there is no relationship between how common a variant is in the population and how big its effect is, so that we under a selectively neutral model, then the cumulative amount of variance should be on this line y equals x. If there's very, very strong selection against effects that are bigger, because they have a negative effect on fitness, then these then we expect that relationship to be following something like this, that we fitness itself. What we find for common traits, and with the caveat that this inference is all from the SNP data, we find clear deviations here from this line y equals of x. And for some traits, you probably can't read all of them, educational attainment is somewhere among all these curves here, but bottom line is there's some evidence for selection, past selection, on loci that are associated with educational attainment. And other groups, for example, the Icelandic group also published on that, even showing that this is not just in a very distant past, but they observe it even over a 100-year time period. And this is the paper which is on bioRxiv, where we actually were interested in detecting the effect of sortative mating. Oh, sorry, no, sorry, I'm mistaken. This actually was published last year. We exploited the UK Biobank, because there's data on spouse pairs. And we essentially looked for whether the phenotypic correlation for a particular trait, like height, was consistent with a correlation between the genetic predictors, because that's what you get when there's a sortative mating. And for height, we showed it very clearly, and we had additional data. But then the referee said, oh, you have to look at some other traits as well. So then we went to the UK Biobank and picked out a bunch of traits, but including educational attainment. So all these traits, for example, where are we? Bone mineral density, we saw it as a null trait. We couldn't see why there would be evidence for assortative mating on loci associated with bone mineral density. although you can always build a story that it should be. Anyway, and we found indeed that's null, but we did find evidence for some of the other traits. In particular, for educational attainment, the phenotypic correlation, which is here, it's 0.4, it's pretty high, is actually smaller than what we predicted from under assortative mating. So I'm not entirely sure the explanation of that, but clearly there's evidence for correlation between the polygenic risk scores of spouse pairs for educational attainment. And of course, we'd love to do this for traits like liability, schizophrenia, but at the moment we're still underpowered there, we think. Okay, this is a slide you can probably not read this at the back, and it doesn't matter. For those of you interested, I just want to have a few slides on what else you can do with these GWAS results when it comes to biology. And the main purpose is to convince you that all these hits that you find in the genome and the genes that are nearby, that it kind of makes sense. So these are all essentially enrichment analysis. So what you can do, you take your 1,300 SNPs and you're saying, what do I know about the region of that DNA? What do I know about it in terms of gene regulation, for example, in different tissues? Or you can say, all those SNPs that have a big effect, what's the nearest gene, and what do I know about gene expression of that gene? Is it preferentially expressed in certain tissues, and so on? So I'm not going to go in great detail to the analysis, but just, I think, comforting, showing you results that, that kind of make sense. So first of all, this is just a schematic of, supposedly, of synapse where neurons communicate. And when we look at all our prioritized genes, and these are prioritized from a whole kind of bunch of different analyses, basically they all seem to point to communications between neurons. So here are all these genes listed, and here's all where they play a role in synaptic plasticity and neuronal communication. So that was kind of cool, so that's maybe what you expect. We can also look at evidence from data which are completely orthogonal to our genome-wide association studies, which is looking at the conformation of DNA, because if chromatin is open, it suggests that you can have gene regulation there. And we found, and this is just again an enrichment analysis, we found an enrichment for gene regulation in fetal brain tissue. Not only in fetal brain tissue, but if you look at the expression of our prioritized genes, they are enriched, so they're more expressed during brain development and all the way from prenatal to adulthood. And that's shown over here in these two lines. These were from our previous publication, and these from our current, the new findings, slightly lower. And here are the null, the other genes that are not prioritized. And when we look at the expression of the prioritized genes, it all points to the central nervous system. Again, to these kind of results that you wouldn't get by chance. As you can see, there is some kind of log p-value here, and you see very, very strong enrichment here. And when we then look at genetic variation in the genome and try to partition that according to, again, by linking it to outside information which tells you where those genes are preferentially expressed, for example, we find an enrichment in the frontal cortex for the heritability explained. And although the standard errors, as you can see here, are quite large. So I hope that I've convinced you that using this kind of big data, and 1.1 million individuals with 20 million SNPs on them is a huge data set, and underlying that with new methods, that we can actually learn a lot about things we studied in the past, but also new questions that we can ask with these kinds of large data sets. And I've given you some flavor of that in terms of nature-nurture questions, questions of non-random mating in the population, getting predictions and testing them what they are correlated with in out-of-sample prediction, putative causal inference about whether perturbations in one phenotype are, whether the data say that perturbations in one phenotype are actually causal with respect to another. I think this is, for the first time, looked in great detail at the X-chromosome, and I'm sure there's more that we can learn there. And also, we can make some inference about past natural selection. And of course, all these methodologies and these kind of data applications can be applied to any complex trait, not just educational attainment, and that includes, of course, diseases and disorders, including psychiatric disorders. Thanks.
Okay. Peter, that was fantastic. It's a bit breathtaking. There's so much happening now. I always got research envy where everyone else's research seems more interesting than your own. I want to do that research. Questions for Peter? So, Andrea first, and then Andrew.
Hi, very interesting. I was interested in the heterogeneity you see in the educational attainment between cohorts. And I wonder if you thought about doing you can do some kind of heterogeneity test across cohort and then see if those SNPs are genetically correlated with other traits to try to understand which other trait seems to be more important in certain cohort compared to other cohorts. Or if you have any other idea why this heterogeneity exists between.
That's a very good question and an interesting suggestion. I think what we're testing with this heterogeneity is partly, I think, kind of scale effects. And they're perhaps not so interesting. You're more interested in something that really, crosses over. So, for example, when I mentioned the effect sizes from our second study in the UK Biobank were almost too good to be true, it's basically because it's such a cleaner data set that the effect size is just bigger because there's less noise. They all have the same protocol. They're all done in the same way. So these things in a number, if you start testing whether the effect size is consistent, like odds ratios or whatever they are in this case, just the regression coefficients, then I think part of that heterogeneity is just scaling effects. I think that essentially you could say within the UK Biobank, this SNP explains more variation than the same SNP explains in a sample from 23andMe, which has a noisier protocol, right? Self-report. Whether you can use this information to identify those SNPs that contribute most to that signal is a very interesting suggestion. I think we need to do that if we were to have two very, very large sample sizes rather than relying on those smaller ones where I don't know what's going on there. There's a lot of noise, but it's a great suggestion.
Sure, I have a simple question. So you showed family history predicted more than polygenic risk. Do they predict anything independently? Like if you stack polygenic risk on top of the family history?
Yes, we have done that. It doesn't explain anymore. You could put it the other way. If you look at the variance explained, if you put them together, if you just look at the variance explained by the predictor, it basically goes down when you add family history. So I have to look it up. Sorry. But interestingly, because in the past, when people have done this, also for height, they say, oh yeah, family history is a better predictor. Well, first of all, there's a few caveats there. That family history could mean the environment of your parents and not just the genetics. So it's not a fair comparison if you then just compare it with a genetic predictor. But even if it were just genetic, then it becomes interesting because the SNPs capture not just the average, they capture some of the average values of the parents, like a half, but they don't capture the segregation, of course. So it must be the case that at some stage the SNP predictors become better than just the phenotypic average of the parents under just a genetic model. And for height, we're very close. It's something like, that break-even point is something like when the SNPs explain square root of half the heritability or something like that.
Naomi and then Espen.
The GE correlation results are very interesting. I was wondering if you could help this group think about how that might impact on the sorts of traits we work on.
Well, I would say the first thing, and that fits in with John's symposium, is just try to get the data where you can measure it. So GE correlation, if it's, as I said, if it's always linked, it will show up as genetic variation. But as we saw, it would mean that there'll be less variation between siblings within a family. I'm not exactly sure what you're getting at, to be honest.
But adoption studies have been used in psychiatry, so that then separates out the potential GE correlation.
Sure. I always thought that adoption studies also have some methodological issues, and that they're usually not large, fortunately maybe, and also that there may still be some correlation between the parents who adopt and these like socioeconomic background of the kids. I'm not an expert on that, but fair enough.
Esper next, and then Jerome.
Yeah. So thank you very much. I learned a lot. So now I wonder. I mean, this X chromosome here. So is it possible to look at the Y? Or is it politically incorrect, or is it just not possible?
Well, there is not a lot of genes on the Y. In fact, the evolutionary geneticists, in particular the females one, predict it's going to disappear over time in the human populations. It might take 1 billion years or so, but it's sort of degenerative. So I don't think there's a lot of evidence. There's a lot of stuff going on the Y, but yes, different. Males will differ in their Y chromosome. So you could look at it if you want. I mean, we haven't done that. There's no reason not to, I suppose. But anybody else know how many? There's hardly any genes on the Y, I think.
But you're excluding the pseudo-autosomal region as well, aren't you? So no one's looking at that.
Well, we actually we sort of reinstated that, because the nice thing about if you have, but it's better to do we're still struggling here with power. When we do this for height, and we're looking at the pseudo-autosomal region, which means there is recombination there, we find no evidence of dosage compensation, as we shouldn't. So that's consistent.
There are around 300 SNPs tagged the UK web and con Y, but it's a big haplotype group, so you should better code the haplotype rather than the person.
Yeah. I mean, traditionally people use the Y for studying population structure and so on.
Right.
Well, actually, just 2 points. Well, the adoption studies are interesting. So I looked into this for schizophrenia a while ago, and whilst the heritability estimates for schizophrenia are robust in adoption studies, it does appear that the actual incident rate of schizophrenia goes down in the adopted away pairs. The heritability estimate remains the same because of the concordances, but the actual rate of schizophrenia goes down, which I think suggests to me that the environment, getting rid of, but it doesn't affect the heritability estimate in some way, so I'm a bit confused by that. So it was maybe 60% of what it should be.
And what kind of sample size is that?
I can't remember now, but it seemed to be a kind of not commented upon conclusion, right, of the studies. And then just the other point about X, right? So X chromosome, I keep telling people this, X chromosome is more important for men than for women, because men have one copy, right?
The rest of the mosaics, the girls are mosaics.
So if you have a female predominant trait, as far as I can see, you should expect a lack of association on X. But a male predominant trait, you should expect a bit more association on X. I interpret that.
Sorry, I don't quite understand the reasoning.
If you look at Mendelian disorders,
well, surely, I mean, you pick up, X-linked, mental retardation because they only have one copy, and if that's the other chromosome can't compensate for it.
I think the same. Yeah, but anyway, I think it's the same logic to some extent. It extends into complex traits.
Well, from the few traits, because in addition to educational attainment, we've looked at a whole bunch of other traits. It doesn't seem to me anything special about the X in that the genetic variation, there's clearly a difference there in males and females. So on the full dosage compensation, if SNP has an effect, it contributes to twice the amount of variation in males than it does in females. So if all the genetic variation for a trait was on the X, and it was all on the full dosage compensation, then you get twice the heritability in males compared to females. But that's not what we observe.
Can I ask Peter the last question? So now I want you to speculate. You're amongst friends. I'll ask David to edit this out from the video if need be. So what are the applications for this powerful genetic instrument for educational achievement, for economics of our nation, for society, for education? Really?
So, I mean, as you showed, I justify working on this not because I want to, want to select, anybody or stream people into different education streams. Partly because the genetics of educational attainment is still the heritability is about 40% or something. So most of it is clearly environmental, right?
People in that lowest decile get double dose education.
Well, I mean, that was the original, from memory, the original justification for when Binet came up with the IQ scale was actually to pick out kids who needed more education. So, yeah. I mean, I think that the applications are likely to be more in economics in terms of inferring causality, which is just as big a problem as it is in medical or epidemiology, because in economics, just like in health policy, if you think that something's causally correlated and you change your policy, and that could be a very expensive exercise in wasting taxpayers' money if the association is not causal. Yeah. And so that's, I think, where I see one application. But also, if these predictors permeate, any disease that involves humans, it just becomes all more powerful, and you can do better quality control. For example, the one I mentioned, where you have 30 university students doing one task. All our colleagues are big on that. Maybe the average predicted value for cognition is just different between the two groups, and it stuffs up your experiment.
We were joking before whether this instrument will predict. Did you vote for the Brexit?
But that will definitely be edited out. Thank you, John.
Before we thank Peter, I actually have a quiz, or I want to ask a question to the audience. We have allocated an hour for lunch, but if we cut that back to 30 minutes and start at 1 o'clock, we can be out of here at 3 p.m, so you can have an early afternoon. Is everyone wanting to have a half-hour lunch? Everyone happy with that? Yay, okay, good. So so Espen, Andrew, Andrea, and Jan will start at 1 o'clock. And could you please maybe compress your talks to 20 minutes, and we'll try to bunch these last 4 sessions in a way that respects the audience's attention span. So you are now free to go for lunch, but I want you now to join with me and actually thank Peter for a virtuoso presentation. Thank you, Peter.